Good morning. Today we are going to talk about nitrate and phosphate transporters. So you know those minerals are absolutely essential for growth, plant growth, and they're needed for DNA, for proteins, and so on, and I'm not going to talk a lot about it. So there are two major transport systems for nitrogen uptake. So first it's low affinity transport that facilitates acquisition of nitrate at the concentration higher than half of millimolar, and high affinity transport, which is important for nitrate uptake under micromolar range concentration in the soil. So what proteins, what transporters transport nitrate? Two families we have already talked about when we talked about anion channels. These are slow anion channel associated homologs and chloride channel. But the main transporters that transports nitrate is nitrate transporter 1 or peptide transporter NPF or NRT1 family, and nitrate transporter 2, NRT2. So that's just to remind you that you see this reverse potentials depending on logarithm of nitrate concentration. It's expression in oocytes of slack channel, and it is linear. So it depends linearly on nitrate concentrations, and this shows us that this transporter indeed transports nitrate, and the same story is for CLS, and the changes in nitrate concentration cause change in reversal potential. So today we will talk about two other families of transporters that transports nitrate and their major channels that actively involved in nitrate uptake in the root and long distance nitrate transport. So NRT1. So this family of transporters is involved in low affinity nitrate transport, except for NRT1. This one is dual affinity transporter, and so it operates at both high and low nitrate concentrations. So overall, there's 23 identified Arabidopsis genes of NRT1, and more than about 130 just in plants. And I just showed this graph. This is just NRT1 type of genes in Brassica and Arabidopsis. So the nitrate transporter was discovered as early as 1993, and one of the authors is Julian Schroeder, that we have talked already about, and they expressed in 1993 the gene in oocytes, and they have seen increased in uptake of nitrate. And this CHL1 is actually chloride resistance to 1 or nitrate transporter 1, NRT1, and we are going to talk about it. So here you see the nitrate transport versus concentration of nitrate in the media, and you see this mutant in this gene has problems in both high affinity and low affinity nitrate range. So it seems that it is involved in both high affinity uptake and low affinity uptake. So this was discovered in 1998, and then in 1995 they showed that it looks like that this is a co-transport, nitrate proton co-transporter. So depending on pH outside, the nitrate transport falls, and nitrate uptake decreases depending on pH in a wild type, and it doesn't in the mutant. So And after certain calculations, they figured out that charge coupling stereometry is 2 protons per 1 nitrogen nitrate. But that is not it. In 2009, they discovered an amazing thing, okay? So they looked at the primary response to nitrate, which is expression of nRT2-1, all right? So this is the first response when nitrogen concentration increases, and this response is two-phase. You see, it has high affinity and low affinity response. So this is not current. This is relative expression of this nRT2 gene, which is a primary response to nitrate. And in the mutant, the one that is null, so the protein is not there, nRT1 is not there. You see that it just flat, all right? But if you take the mutant that makes a whole protein, but with a mutation that makes it incapable of transporting nitrate, all right? You see, the primary nitrate response is fine. So this is CHL19 and no problem. Absolutely, it's still initiates response, a primary response to nitrate. So basically, what it tells us that nRT1 does not need to transport nitrate into the cell to transduce signal, a nitrate signal. Therefore, it's a bona fide receptor. Okay, so it interacts with nitrate and initiates signaling in the plant that is signaling in the plant that is important for the for uptake and assimilation of nitrate. So actually nRT1, it governs a lot of different responses, including primary nitrate response, also longer term transcriptional changes, of course, and root growth and development, and we are going to look at it, germination, flowering, and sensitivity to various abiotic stresses. And these all responses are due to its receptor function. So for example, here, you see the, also this protein, this is the structure, 12 transmembrane domains, and this is Reunion 101, which is phosphorylated. And if this amino acid is mutated, this is CL19, and it renders channel non-functional, so it does not transport nitrate anymore. So in case of when you express in the mutant the protein with where this phosphorylation site is mimicked as always phosphorylated, what you see is the protein retains high affinity response, you see? This is again relative transcription of nRT2, so it's primary nitrate response, okay? So the high affinity is there, low affinity is not there. So that's what phosphorylation does. And in this case, it's dephosphorylation. So this one mimics always dephosphorylated threonine. And interestingly, it's higher, response is higher. So the uptake, the relative expression of nRT2 2 increases. And here I will show you the response of plant roots, lateral root development in roots to nitrate transceptor. So here you see density of lateral roots and in 0 nitrate. So in wild type, the lateral roots do not develop in the absence of nitrate, which is logical, right, for the root. And the only protein that acts the same as wild type is always phosphorylated, NRT1, whereas incapable of transport, null mutant, and dephosphorylated, they don't. And they are the same in high nitrate conditions. So basically, here in this case, for this response, we know that phosphorylated form of NRT1 is responsible for regulation of lateral root development. But the amazing story doesn't stop here. Apparently, when they expressed this transporter in oocytes, they discovered that this transporter transports not just nitrate, but auxin. This transceptor, sorry, it's transceptor now, auxin. So here you see the uptake of auxin in oocytes depending on nitrate concentration. So when nitrate is not present in the media, it transports auxin, see? And the higher nitrate, the less auxin is transported. So they're competing for it. That's what it looks like. But auxin itself, if you increase auxin concentration outside, it does not affect nitrate transport by this transporter. Again, there is so if there is a mutation in NRT1, which is CHL15, you remember it's a null mutant, then in nitrogen-free media, you see decrease in the plants in auxin uptake, whereas in a higher concentration, it doesn't happen. You see? So this suggests that what we have seen when we looked at the expression in oocytes, it's also true in plants. So when there is low nitrate in the media, then the transporter is capable of transporting auxin into the plant. Also, this is the 2,4-D uptake, which mimics auxin uptake in different mutants of NRT1, mutants that we have talked about. Again, xenopacyl injected with RNA. And what we see is the NRT1 wild type and phosphorylated mutant, always phosphorylated, ensure the increased concentration of accumulation of nitrate. So this is kind of model of how NRT1 mediate biphasic control of nitrate signaling. You remember we had the primary nitrate response had this biphasic shape. So it is biphasic. So the idea is that at low nitrate concentration, so the nitrate binds only to the high affinity site of monomer, which initiates priming of threonine 101 site for phosphorylation by CYP and 23 and calcineurin. Nice. So this is phosphorylated, and once it's and this basically turns it in high affinity nitrate transporter. And in higher concentration, both of monomers bind nitrate, and that maintains dimeric state of NRT1 by inhibiting CYP23 kinase. So this turns it into a low affinity. So that's what current hypothesis is, but there are certain things that haven't been proven yet. Okay, NRT2, it's nitrate transporter 2. That's how it looks, and 7 proteins have been of this family have been identified in Arabidopsis. So high affinity root influx is mediated by NRT21, 22, 24, and 25. So they play a key role in a lot of biological processes, including nitrogen starvation, again root architecture, seed development, cadmium uptake, and plant microbe interactions, and maintaining appropriate carbon-nitrogen ratio. So these, here you see the nitrogen uptake and versus external nitrogen concentration, and this is a uptake in wild type, and this is in the mutant, and you see the high affinity, which is in micromolar range, is affected, whereas in high affinity range, when external nitrogen concentration is in millimolar range, they're fine, more or less. Okay, so what roles they play? They are involved in seed development, so transport of nitrogen into the seeds, developing seeds, translocation and remobilization of nitrogen, and of course high affinity transporters are involved in root mediated uptake of nitrate. So this, now I will tell you about the current model of regulation of NRT2 transporters. So it all starts with the synthesis of short peptides by root cells that are starved by nitrogen. And these peptides are transported to the shoot. Here they are, and through that xylem they are transported to the shoot. Then in the shoot, they're being recognized by a receptor, CEPR1. And this receptor translates this CYP signal to produce glutaredoxin polypeptides, so certain polypeptides. Here they are, these polypeptides, and they move back to the root, and they induce transcriptional response, and they upregulate NRT2 and CEPH. Here it is. So what CPH does? What CPH can do is selectively dephosphorylate NRT2 at serine 501 residue, but you don't have to remember these details. And dephosphorylating rather than phosphorylating, in this case, dephosphorylating restore high affinity transport into the cell. So that's how nRT2 is regulated. Okay, now we are going to talk about the main nitrate transporters that involve the nitrate uptake, transport, and remobilization. So so to begin with, the most important function is acquisition from the soil into the root. It is nRT2 1, 2, 6, 4, 5, and 6, 3, and 1, 1, sorry, or nRT 1, 1 or NPF 6, 3. Don't be confused by all these different names of the genes. The thing is that people discover the gene and they name it a certain way, and then they're discovered via another mutant screen and by other people, and they call it differently. So which one is used basically depends on researcher. All right, so these are high affinity transporters that take up nitrogen from the soil. And there is one that performs NPF 2.7 that performs efflux function, so loading of the phloem. So the nRT2, 4, and 5, they are important not just in uptake but in root to shoot transport, because that's another important thing to transport acquired nitrogen to the shoot. So xylem loading and unloading. So nRT 1.5 is involved in xylem loading with the nitrate, and 1.8 and 1.9 are involved in xylem unloading, which is important. And nRT 1.6 and NPF 5.5, they act as remobilizers. So basically, you store nitrogen in the vacuum, you store nitrogen, and then you have your seeds develop, and nitrogen is really needed for the seeds, and so you remobilize it from other cells, other tissues, and you transport it, the plant transports it to the developing seeds or developing younger leaves and so on. So that's basically nitrogen transport for you. Plants do need nitrogen. So the next story would be about phosphate transporters. You understand that phosphate is just as important as nitrogen for plant growth and development. So there are also a lot of transporters for phosphate. But basically bear in mind that this research, understanding of transporters of phosphate and nitrogen transporters are exceedingly important, because at the moment we fertilize the agricultural plants with nitrogen and phosphorus fertilizers, and you probably heard that it's very bad for ecology and it's very bad for the soil, and it is indeed. So, and it's going to be worse. And optimizing phosphate and nitrate acquisition and storage in the plant is very important, because at some point we will be faced with the deficiency of these fertilizers and inability to use them, because they really spoil the soil. And so we will, in order to engineer agricultural plants that will be efficient in the uptake of these nutrients, we really need to understand how these transporters function and how the transporters function in model plants to maybe make transgenic agricultural plants with desired qualities, with desired efficiency of the uptake of phosphorus and nitrogen. So in 1990, it was discovered that uptake of phosphorus from the soil is actually energy dependent and makes use of proton gradient across the plasma membrane. So here is our net concentration of change in phosphorus and protons, and concentration of protons is in pH. So what would you guess from this graph? Again, it's so again, it's 1990. We didn't have any genes for phosphate transporters. They had no idea how phosphate is transported. So what they looked at is they added phosphate, and they looked at what happens with external pH and phosphorus concentration external. So it's an easy, relatively easy experiment. You see what I mean? So what would be your conclusion from this?
The phosphate is transported into the cell. That is why it is this concentration decreases, and their proton concentration decreases as well. That is why their pH increases.
Yeah, exactly. So from this experiment, the relatively simple experiments, they came to the conclusion, it is likely that it is a co-transport, okay, of phosphate and protons. Then in another paper, they looked at effect of phosphate on cytosolic pH and membrane potential. So, Sash, what can you say from this? So phosphate is added here.
Yes, we can see that when we add phosphor, the membrane
transiently, yeah, depolarizes.
Depolarizes, but then it's hyperpolarizes again.
When the phosphorus is taken out. You see, it's a system where you transfuse water, it's a solution, okay, through the chamber with the root. So you add a certain ion and then you can remove it. You understand? It's like a river.
Mm-hmm.
So yeah, so basically the important thing is that when you add phosphate, the pH, cytosolic pH,
it decreases.
it decreases.
Which indicates that there is a higher proton concentration in the cytoplasm, and we see that when phosphorus is taken out of external environment, cytosolic pH is restored. It becomes more alkaline.
Cytosolic becomes more alkaline. No, pH outside becomes more. This is pH outside and this is inside.
But when
You can see that pH increases when phosphorus is taken out.
It's not out.
Out.
Yeah.
So when they were adding phosphorus, cytosolic pH decreased. When they removed it, it increased again.
Yeah.
That's fine.
So that's the first experiments which showed that it is that phosphate is taken up by co-transport with the protons and using the energy of proton gradient. So in 1996, they have discovered, they have cloned the PTH family of phosphate transporters. And here is hydropathy plot. So these are hydrophobic regions. So it's transmembrane regions. And here's Arabidopsis PT1 and Arabidopsis PT2. And they have shown that it is indeed a phosphate transporter. And they discovered it by complementation of yeast high affinity phosphate. That, by the way, was a very common way of finding transporters. So you have there is a big library of yeast mutants that would not, say, grow on low phosphate because they don't have a certain transporter. So you can grow them on higher phosphate and propagate them, and then you take them and put them in low phosphorus after transforming them with cDNAs, a lot of different cDNAs, all right? So you take cDNAs from Arabidopsis plant, say that I expressed at phosphorus deficiency conditions, right? So you grow Arabidopsis for low phosphorus. You grind it up. You get RNA. Then you get synthesized cDNA. Then you make lots of plasmids with this different cDNAs. So the only one, and you transform yeast mutant that does not have this phosphate transporters with these cDNAs. And the idea is there's lots of them. You don't know exactly which ones. And then you plant this mutant that is incapable of growth on low phosphate and low phosphate. And then you get colonies that grow on this. And that's how you look at it, and this gene must be involved somehow in phosphorus transport. And that's how in 1996 that they actually discovered this transporter. And you see with just the vector, it's not, the yeast doesn't grow. You see its optical density, basically growth of the yeast. And with both PT1 and 2, they are growing fine, so they are able of transporting phosphorus. So the phosphate transporter family, this PTH, that's the main family that is involved in uptake of phosphate. They are grouped in several types based on their cellular localization and basically sequence identity. So the main is PTH1, and here it is. It's sequence 12 transmembrane domains and big extra and intracellular sites. And here is the cell with the cell wall and chloroplast and mitochondria and Golgi and vacuole. And you see all of them have their own favorite transporters, phosphorus transporters. And PTH1, it is mainly in plasma membrane. PTH2 is in chloroplasts, and PTH4 is in chloroplasts. And PTH3 is the smallest one, is in mitochondria, again transport of phosphorus in mitochondria. PTH5 is specifically in the vacuole, and 4 also in chloroplasts. We have already talked about it. It's in chloroplasts, and it's in Golgi, which is very important, and it's also in non-photosynthetic plastids. They're not here, but they're not presented on the figure. So the main transporters, phosphorus transporters, that maintain phosphorus uptake in the roots are PTH1,1 and PTH1,4. So how do we know? And here they are. So how do we know it? It's because if PTH1,1 is mutated, then uptake efficiency falls 30%. And in double mutant one, PTH1,1, PTH1,4, it falls 60% in double mutant. So these are important. So I will tell you a story about regulation of PTH1 transporter as an example, because all these transporters are under very scrupulous and complicated regulation on all different levels, including chromatin level, transcription level, translation level, post-translation translation level, and so on. So, so this is our PTH1, high affinity transporter that that is responsible for a major part of phosphorus uptake. So its activity has to be tightly regulated. To begin with, there is ARP6 and IPK1 that are IPK1 and ARP6. They deposit H2AZ, so-called histone variants on chromatin. So, and this variants inhibit PTH1 expression. So they deposit this variant at the part of the chromosome where the gene is. So it inhibits the PTH1 expression. So that's what happens at high phosphorus, when concentration of phosphorus is high in the plant and outside. So we don't need to use a lot of energy to uptake and get more phosphorus. 2 transcription factors, so of Werke family. So Werke 42 upregulates expression of PTH1 under deficient conditions, so it increases expression. Actually, under sufficient conditions, so that's what it does. Whereas 45 regulates at again at expression of PTH1 at under phosphorus deficiency. So these are important. And there is also another Werke transcription factor, which is Werke 6, and it is interesting. It inhibits expression of PTH1 in response to high arsenic levels. So in the soil, so why specifically arsenic? Because actually this transporter cannot help transporting arsenic as well as phosphorus. So if arsenic level in the soil is high, we really don't need a lot of PTH1 because it will poison the plant, because it will be a gateway for arsenic into the plant, which is not really good. So there is a specific transcription factor that is taking care of it. So PHR1 is the major protein, major transcription factor that is responsible for the phosphorus status of the plant. So there are a lot of different proteins that regulate it, and it regulates expression of our transporter. It's called phosphorus transformation response 1. So how it is regulated? So first C1, which is small ubiquitin like modifier, SUMA, E3 ligase induced simulation and activation of PRH1. Have you heard about SUMA, simulation? Good. So it's simulated and when it is simulated and activated. You remember E3 ligase, what does it do?
It binds the ubiquitin to the protein to
It's not ubiquitin. It's simulation.
then it is SUMA protein which is binded to the protein, and it is somehow regulated the fate of this protein.
Yes. So what protein recognizes its target? Yes, there is E1 and E2, but E3 is often a receptor because it actually recognizes the protein that it needs to be ubiquitinated or simulated in this case, right? So it's specific. So C3 induces simulation and activation of PRH1. This is all transcription. This we are talking about transcription. And SPX1, that is a nuclear protein that is induced by starvation. It antagonizes the action of PHR1, so here it is, and thus it interacts physically with PRH1, so it inhibits binding of PRH1 to target gene promoters, and you see PRH1 is regulating quite a bit of different genes. So this thing inhibits PRH1 by just binding it, physically interacting with it. Okay. All right, so now we are getting into post-transcriptional level. Post-transcriptional level. So the PRH1 upregulates microRNA 399. And this is actually quite cool because if you have this expression of MI399, it can travel through the xylem to other parts of the plant and induce the low phosphate response. And it's this microRNA induces 4-2 cleavage, and 4-2 inhibits the PHT1. You see? So it inhibits an inhibitor. All right. So it activates basically. By cleaving the RNA of 4-2 that inhibits PHT1. So this IPS7 is non-coding RNA that regulates level of this microRNA 399, which is again not only activates this expression of this levels of this transporters, increase level of this transporter, but also signal travel through the xylem to signal other parts of the plant that phosphorus is scarce. Right, there is another microRNA that is induced by PHR1, this major regulator, and it cleaves the transcripts of NLA, nitrogen limitation adaptation, NLA protein. It is again E3 ligase that controls ubiquitin dependent degradation. So normally this NLA degrades pH T1. So it's synthesized and degraded, because ubiquitin is added to it. And NLA inhibits this process. So why do you think they need a basic number of PTH1s in the cytoplasm that is synthesized and degraded? Even if there is no starvation.
Because it is an energy-dependent process, so we should always keep the energy and the balance.
To make sure that if no phosphorus, it will get into the membrane, right, and start functioning. Okay, so now we get into post-translational level when we have protein there. So this PHF1, it's again a starvation-induced factor, and it causes trafficking of this PHT1, actually not just 1 but 2 and 4, from ER to the plasma membrane. This is important. So they may be in ER, but when it is when you get a signal of low phosphorus, all of these them are delivered to plasma membrane to function. So here it is. So this trafficking of PTH1 is blocked when when it is phosphorylated by casein kinase. So this is certain kinase. So you see the regulation of PTH1 transporters is very complicated. It's on many levels. So, and it's probably ideally adjusted to different phosphorus levels, to respond to different phosphorus levels and have appropriate concentration of the transporters inside the membrane. Here you see the recordings of phosphorus-dependent current in isolated vacuoles, and so this shows the phosphate influx, and you see in the mutant of PTH5, it's seriously inhibited, so there are PTH5 is localized to the vacuole, and it is indeed ensuring phosphate transport into the vacuoles. So other phosphate transporters families, so the 4-hydroxylase phosphate 1, it belongs to certain superfamily and localized to the Golgi and to the trans-Golgi network. So the idea is that it plays a role as a phosphate transporter. However, there is no currently clear evidence that it actually transports phosphorus. Another 4-1 homolog involved in transferring phosphorus from the seed coat to the embryo in developing seeds. Another members, this is rice transporter SULTR, 3 and 3 and 4 are also likely involved in phosphorus accumulation in rice grains. However, no detectable activity for phosphate when tested in yeast and xenopasoocytes. Again, I just want to point your attention that this is not an absolute proof that it's not working as a phosphate transporter, because extracellular environment in plant cells, in yeast cells, and in xenopasoocytes are so different. So maybe it is activated by some factors in the cell wall, all right, in plant cell wall. So when it's working, when transporter is actually active in oocytes or yeast, it pretty much proves that working in plants. However, it's not the other way around. Also, there is salt 3,4 that also transports phosphate. Okay, so this is just the same scheme you've looked at. It's potentially 4,1 is potential phosphate transporter, and again there is no direct measurements of phosphorus transport by it. All right, so now we will be talking briefly about membrane pumps. So pumps, active transport, it uses proteins and pumps to concentrate molecules against its concentration gradient, and that's why plant cells have membrane potential, and that's why life is possible, pretty much, and membrane transport very much depends on it. So three types of ATPases, P, V, and F types. ATPases, and the P type is very widespread family, and for example, sodium potassium pump that is in our plasma membrane is also P pump. It's a signal large catalytic synthetic subunit that is inhibited by vanadate, which is very useful, and it's pump irreversibly and all of them pump cations. So this would be the structure of plasma membrane ATPs. So huge cytosolic domain, 10 transmembrane domains. We have actuator, so-called domain, nucleotide binding domain, and phosphorylation domain. And when ATP binds to N domain, then it moves closer to the P domain, and it forms catalytic site. So this is AHA2 of Arabidopsis thaliana. So how they are regulated? Of course, regulated is very elaborate and on all different levels, just like phosphate transporter, they're more complicated, but we don't have time to discuss all levels of regulation, so I will tell you about regulation of proton ATPs by phosphorylation. So proton ATPs have very many sites that can be phosphorylated. So what we know about is different environmental signals induce phosphorylation of proton ATPs at different sites. For example, peptides, Ralph, we will be talking later about it as well. So they activate multiple protein kinases that will phosphorylate it on S890 on this phosphorylation site. Okay. So, and that would inhibit its activity. See? This phosphorylation will inhibit its activity, whereas other environmental signals will induce phosphorylation on 881, which activates it, right? Other can induce phosphorylation and 947, which would inactivate it. And others, also 1433 proteins are very important, of course. They bind to the proton ATPs and activate it. So, and for example, alkaline stress will induce inhibition of proton pumping activity by phosphorylation and releasing 1433 protein. So, plant hormones also regulate proton ATPase through phosphorylation. For example, auxin induces sour proteins, a lot of them. It's first response pretty much to auxin. You add auxin, you get a lot of sour SAUR proteins in the cytoplasm expressed, synthesized. They block protein phosphatase 2 that dephosphorylate tyrosine at 947 and inactivates it. So it will do what? So it activates the one that blocks, so it activates. So would an auxin inhibitor activate protein ATPase here? It will activate because it activates inhibition of inhibitor. All right? Okay. And brasinosteroid is working in a similar way, but through BRI. It also affects this phosphorylation site. All right, other stimuli from that reflect the condition of the plant. So light causes expression of SOSUK2, which will cause this phosphorylation. Then you get sucrose that induces this phosphorylation, and the high ratio of carbon to nitrogen, so low nitrogen, high C, will affect association of 1433 protein with ATPase, and of course it is by no means a complete number of factors, environmental factors and hormones that affect activity just of this ATPase, AHA2 ATPase. So, yeah. Also, the target of oxidative inhibition by ROS. So, you know, all the stresses, pretty much all stresses, cold stress, hot temperature stress, pathogens, you name it, all stresses for the plant associated with this increased activity of reactive oxygen species. So NADPH, the membrane activates and so on, and so you get increased reactive oxygen species, and immediately reactive oxygen species such as hydrogen peroxide blocks ATPases on the plasma membrane. Okay, so this is a P-type proton ATPase. So another ATPase that is very important is calcium ATPase. You remember we talked about calcium being 10,000 times less in the cytoplasm than in the outside media, so we need to pump it all the time, right? So there is ECA and ACA type calcium ATPases. So ER calcium ATPases, and there are 4 of them in Arabidopsis genome. They transport calcium two calcium ions per ATP, and they have rather short C and M terminal sequences. And ACA is what type 2B. They're called plasma membrane calcium ATPs, and there are 10 members in Arabidopsis, and they transport 1 calcium per ATP split and have big autoinhibitory domain. So this classification came to us from studies in animal cells. They also have these 2 types. So type 2A. So what is the function? Okay, I will show you some mutants of calcium ATPases, but I just want to mention that there are a lot of calcium ATPases. They're very important and they're redundant, of course. So if you get rid of all calcium ATPases, the plant will not, embryonic cell would not divide, right? It's clear that they're very important and they do substitute each other. But for example, for this ATPase, the Arabidopsis EC one mutant. They grew really not very well on calcium low media, so the idea is that pumping is calcium by AC1 into ER is important, but we don't know why. Okay, so type 2b, that's how it looks. 10 transmembrane domains and ATP binding site and autoinhibitory domain. And in animals, this type of calcium pumps always are in the plasma membrane. And a lot of different factors also regulates its activity, including calmodulin binding, which makes it active, and localized membrane calcium hotspots, and acidic phospholipid binding, and so on. So it's just to compare plant calcium ATPs of this type and animals calcium ATPs of this type. And plasma membrane calcium ATPs in plants are localized in plasma membrane and ER in tonoplast, plastid envelope, and nuclear envelope, whereas in plants, in animals, it's only localized in plasma membrane. They both have calmodulin by dependent regulation. And autoinhibitor domain is located on N-terminal in plants and on C-terminal in animals. So this calcium ATPases, these calcium ATPs are really important in decoding calcium signals. So we talked about calcium channels that open in response to certain stimuli, and their opening time varies depending on what channel it is, what signal it is, and so on. So you have different calcium signatures in the cell from different signals. And I just want to point your attention to the fact that calcium ATPases are just as important. As soon as calcium concentration increases in the cell, calcium ATPases started to pump it out. And rate with which it does it, and how quickly it is switched on, and how quickly it gets rid of it, you get different calcium spikes, calcium signatures, that is decoded by different calcium binding proteins, kinases, and so on. So you get calcium increase, calcium binds calmodulin, calcium calmodulin binds AC, and multiple calcium activated protein kinase can inhibit it and activate it. But calmodulin needs to be bound, and serine 22 needs to be phosphorylated for this ATPs to be active. So the functions are, again, functions are very important. We don't know, we know only a little fraction of it. So we know Arabidopsis AC9, for example, is required for normal pollen tube growth, and they are important in the response to salt stress and pathogen attack. So this is 2B. All right, so there is also V type of ATPases, which is multi-subunit ATPases, and that's how beautiful they look. And they consist of two domains, formed at least by 13 different E subunits. Membrane peripheral V1, which is AA and AH. And you have, this domain is responsible for ATP hydrolysis, and membrane integral V0 responsible for proton translocation. So this type of ATPases, they are on the vacuoles, the permafrost, and the acidified region of Golgi, TGN, late Andersons and vacuole. So here they are, and they're also very important. Their activity is modulated by hydrogen peroxide and nitric oxide, and they're inhibited by this reactive oxygen species. Ethyl malmate, oxidized glycosine, thyroidoxine, and H2S. So this figure you have already seen. So reactive oxygen species inhibit not only proton ATPase on the plasma membrane, but proton ATPases on TGN and vacuole. So there is another type called F-type ATPase, and we are not going to talk about it because ATPases are multi-subunit enzyme complexes, and they are found in anaerobic transition membranes in eubacteria. They basically mitochondria and chloroplasts, and they role is synthesize ATP from ADP and phosphate under abiotic condition, aerobic conditions using protein motive force generated by respiration as a source of energy. And that is it for now. Thank you very much. Так, теперь у нас есть немножко времени для вопросов.
Ну, значит, у нас был вопрос о том, как, собственно, что собой представляют вот эти аминокислотные замены, какова их функция. И так, значит, замена вот эта замена в CPK24 и в CPK11, она ингибирует киназную активность этих двух киназ. И здесь мы видим, что когда, собственно, когда обе киназы активны, у нас ингибируется активность канала SPIC, а когда хотя бы одна из этих киназ ингибирована постоянно, то мы видим, что активность канала SPIC не ингибируется. То есть это говорит о том, что для того, чтобы ингибирование канала SPIC состоялось, нужно, чтобы киназная активность была у обеих этих киназ. И также был получен мутант CPK11, который конститутивно активен. И здесь мы видим, что экспрессия канала SPIC с конститутивно активной CPK11 тоже не ингибирует напрямую канал SPIC, что говорит о том, что, собственно, CPK11 не ингибирует непосредственно SPIC, а для того, чтобы делает это через киназу CPK24.
Меня спрашивали рассказать подробнее о механизмах потенциал чувствительных красителей. Собственно, вот они. Первый DI4 ANIPS. Это потенциал чувствительных красителей делится на быстрые и медленные по времени окрашивания. Медленные — это за секунды и больше, тогда как быстрые — это, понятно, меньше. Данный краситель относится к классу быстрых, и его механизм работы — это при изменении мембранного потенциала у него происходит перераспределение заряда, и он меняет свою окраску. Собственно, электрохромный механизм. DBAC4 относится уже к группе медленных красителей. Вот. Для того, чтобы он сработал, требуется несколько секунд. Его механизм работы заключается в том, что это заряженная молекула, и мы видим, она довольно гидрофобная, она перемещается через мембраны в зависимости от изменений потенциала, потому что, поэтому, собственно, здесь, собственно, картинка была вставлена исключительно для того, чтобы показать, что мы видим, что когда у нас понижается потенциал, у нас красители аккумулируются в клетках, и она начинает, собственно, светиться, флуоресцировать и становится видимой. На этом все. Спасибо за внимание.
Вопрос заключался в том, почему исследователи используют именно мутанта глабра-1 в качестве, скажем, некоторого контроля при изучении транспорта ионов и засоления. И вот исходно авторы утверждают, что потому что у нас этот мутант не влияет на чувствительность к засолению, в отличие от контрольного растения дикого типа. И авторы ссылаются на два источника, в которых упомянута ровно та же формулировка. Что мы видим? Авторы этих статей, про которых мы с вами говорим, пишут ровно ту же формулировку. И, соответственно, автор, который публиковался позже по времени, ссылается на исходного автора. Мы идем к исходному автору, видим опять же эту формулировку и видим, что он вообще ни на кого не ссылается, и, соответственно, эта формулировка взялась, возможно, из ниоткуда. Я залезла в источники, которые указывает автор этой статьи, почему, вообще, что он использовал в качестве литературы. Там была единственная статья, посвященная мутанту глабра-1, и, в принципе, эта статья вообще ничего не говорит про чувствительность к засолению, там слово засоление не упомянуто даже. То есть мы видим, что про что-то они рассказывают, то есть это практически исходная статья, посвященная изучению мутанту, и, в принципе, все. Я попробовала поискать другие статьи, возможно, это откуда-то взялось в другом месте, и мы видим, что теперь уже авторы другой статьи утверждают, что использование глабра-1 в данном случае к плесени, вообще грибковому заболеванию снижено. В принципе, это хотя бы можно объяснить тем, что, возможно, изменение как раз-таки свойств эпидермы может этому способствовать. Но, опять же, в принципе, как бы и все. То есть именно про засоление и влияние засоления на глабра-1 мы с вами не видим. Я нашла еще пару статей, в которых обсуждается влияние засоления, но вообще на другие факторы, которые участвуют в развитии трихомы эпидермы. Соответственно, мы из этого выясняем, что это утверждение взялось из ниоткуда. Вот на этом все как бы.
Большое спасибо.